Hey, Hammy here, back with the part one of section 6.2, in which we will look at some of Mendel's crosses and we'll explain them in terms of the math and stuff that he used in order to help explain it, and also the idea of Punnett squares and how we can use them to illustrate Mendel's crosses. Okay, one of the first things we'll have to look at is this idea of probability. Okay, that helps explain some of the outcomes of his crosses. Okay, probability is the likelihood or the chance that an event will occur. Because remember, we said Mendel was very smart and studied science, but also had kind of that mathy brain. So he was able to put some of these numbers and see some patterns in his crosses. Okay, now the formula that we use for probability, for example, okay, if I take this nickel here, okay, what's the chance if I flip this nickel that I get heads? Okay, well, the probability is going to be equal to the number of chances for that event to occur. So how many heads are on this nickel? I'll let you see it close up here. It's not a trick nickel, okay? There's heads, there's tails. Okay, so the number of chances is 1. The chances of getting heads, there's only 1 heads on this coin, out of, okay, the total number of possible events. Okay, total number of possible events. Well, if I flip this coin, how's it going to land? One side or the other? Assuming that I'm not some trick magician and can get this to land on end. Okay, it's going to land on either side eventually. So 1 out of 2 chances. So when I flip a coin, what's the chance of getting heads? 1 out of 2, or 50%, or 50-50. What about if I flip it a second time? 50-50. What about a third time? 4th time, 5th time, 6th time? Every time I flip this coin, no matter if I got heads 5 times in a row, well, I'm about due for a tails. No, every time I flip this coin, it's always 50-50. It's always 1 out of 2 or 1/2. Okay, statisticians call this the rule of independent events. Every time I flip this coin, it's a 50-50 chance. Okay, so every time 2 things cross, genetics, there is the same probability that those genes will separate into gametes, and any of those gametes could combine an equal chance each time. Okay, so that's just kind of the basic laws of probability. One more thing I should mention is what we call the product rule. Okay, because remember, Mendel followed more than one trait at once. So what happens if I take 2 coins? What happens if I take a penny and a nickel? Okay, what's the chance if I flip 2 coins of both coins landing on heads? Okay, well, what we do is we use the product rule. Now, the product rule is, when you think of a product, it's something you multiply. So if you look over here, the penny, here's heads, here's tails. Okay, heads, tails. They can, when I flip the penny, it has a 50-50 chance of being heads or tails. So the penny has a 1/2 chance of being heads or tails. You look at the nickel, same thing. Here's heads, here's tails. So the nickel has a 50-50 chance. Now, look at all the different combinations. You can have heads on both, heads on the penny, tails on the nickel, tails on the penny, heads on the nickel, or both tails. So we actually get the total number by product by multiplying. 1/2 times 1/2 is 1/4. Okay, so the chance of 2 coins both coming up heads is 1/4, or about 25% chance. Okay, and that becomes important when we look at doing the dihybrid crosses. Now, when we actually start looking at some of Mendel's crosses, it helps when we use these little squares called Punnett squares, and we've kind of been using them some already. The Punnett squares is named after geneticist R.C. Punnett, right around 1900s, okay, early 1900s. This was when Mendel's work was being rediscovered, and the university that he was teaching at, he really sort of put forth, actually wrote kind of a textbook called Mendelisms, that he really began to explore this idea of doing genetics. And so he used these squares to kind of help organize what's going on here. So if we look at Mendel's monohybrid cross, and let's just use tall and short, for example. And so remember, we're starting with the pure lines, or true breeding. So pure tall would be homozygous dominant, pure short, homozygous recessive. Okay, on the outsides of the Punnett square, we're going to put the gametes. In other words, this pure tall, what gametes can this make? Well, a big T or a big T. That's going to pass on a big T, right? And the pure short plant will pass on a little t or a little t. Those are the two options up here. And now we just combine them. We always put the dominant allele first, big T, little t, big T, little t, big T, little t, big T, little t. Okay, so inside the Punnett square now are showing all the possible F1 gametes. Okay, so these are all the possible F1 gametes. Now, we remember when Mendel crossed purple and white flowers, what were the F1? Okay, they were all purple. So when we cross tall and short, and tall is the dominant trait, the genotype, look, they're all the same. They're going to be 100% big T, little t, which, if you remember from the last video, is heterozygous. Okay, phenotype, physical appearance. How are they all going to look? They're all going to be tall. Okay, so the F1 are all going to be heterozygous tall. Okay, in the next step, we're going to take the F1, big T, little t, and we're going to let them self-pollinate, okay, or even cross with each other. It doesn't matter because they're all the same genotype. And when we look at the possible F2 offspring, okay, we're going to, what can they pass on? Well, then now they can pass on a big T or a little t. The other one over here, same thing, big T, little t. That's what it's going to pass on. Remember, the gametes are on the outside here. So then we just combine them, big T, okay. Again, remember, we keep the dominant allele first. Big T, little t, little t, little t. Okay. Now, the genotype ratio, the genotypic ratio is a little bit different. Okay, here we have tall, 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 short. Okay. Now, not all the tall ones have the same genotype. This one, the first one, is going to be homozygous tall. Okay, to 2 heterozygous tall, to 1 homozygous recessive. Okay, so we say the genotypic ratio of the F2 of a monohybrid cross is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive. Now, how do they look? Physical appearance. How many tall are you going to have? 3 to 1 short. Okay, and we've seen this ratio before, right? 3 tall to 1 short, and that's our 3 to 1 ratio. That's actually a phenotypic ratio.